buenas tardes a todos. Realmente es un gusto enorme que esté participando en el workshop Eva Dyer. Ella se formó en la Universidad de Miami, donde hizo sus estudios iniciales. Luego obtuvo su PhD en ingeniería y de computación, eléctrica y de computación, en la Universidad de Rice, y en este momento se desempeña como profesora en el Culture Department de la Instituto Biomédico, el Departamento Biomédico Departamento de Ingeniería Biomédica del Instituto de Tecnología de Georgia, de la Universidad de Emory. Eva trabaja en lo que sería justamente lo que estamos tratando de hacer en este momento, que es acercar las neurociencias y el aprendizaje por computadora, desarrollando aproximaciones computacionales para interpretar datos de neurociencias complejos, así como diseñar distintos modelos de inteligencia por máquinas inspirados en los modelos biológicos, justamente lo que sería una ingeniería reversa, ¿no? A Eva yo la conocí, tuve el gusto de conocerla el año pasado en un workshop de aprendizaje profundo, de técnicas de aprendizaje profundo para el análisis de imágenes por microscopía en el Marine Biological Laboratory en Goose Hall. Y bueno, cuando organizamos este workshop pensé que era la mejor opción invitarla a ella para justamente acercar estos temas que queremos nosotros tratar de promover a través del workshop y, bueno, eso, ¿no? El acercamiento de la inteligencia artificial, el machine learning, a las neurociencias con la mutua inspiración. So, Eva, thank you so much for being here. It's a pleasure, and please share your screen.
Okay, great. Thank you. Thank you again, Maria, for the invitation. And looking forward to. So yeah, I mean, I know you said that, you know, in terms of questions and whatnot, you know, you didn't want to or the translations, but yeah, if there are questions that you think are good in terms of clarification, right, before I move forward, and I'll try, I speak very quickly. So if there's anything you want to give me as feedback, feel free to stop me. So,
acá, además de nosotras que vamos a estar muy atentas a la presentación, Zaira will be helping with the translation. So thank you, Zaira.
okay.
Zaira Cancela is working also in the laboratory. Will help
great.
us.
So you can see my screen, okay?
Yeah.
Okay. Excellent. So, so I will talk today about work that we're doing in my laboratory to essentially find representations that are embedded within activities of neurons or within neural activity that's collected in the brain, and trying to understand and compare those representations that are being formed by many neurons simultaneously, and comparing those recordings across, as I said here, time, space, meaning different neurons potentially, and also across different behaviors. So this is really about, you know, how do we take neural recordings that are very high dimensional and find low dimensional descriptions of them that allow us to more succinctly represent the data, and then find these comparisons as well. Okay, so a lot of the motivation behind the work in my laboratory is coming from sort of the advances over the past decade or so within data collection, in particular within neuroscience. And so we've now developed these tools that allow us to, you know, take pictures of the brain at very high resolution, both in time and in space, meaning seeing, you know, individual neurons and being able to see many of them simultaneously in order to, you know, obtain a picture of how collections of neurons are working together. And from all this data, we're often faced with a lot of challenges of interpretation and really trying to understand, if we're not looking at the brain at a single neuron perspective and we're looking at how many neurons are working together, we need more efficient descriptions of that information. And so one of the key things that we're interested in doing in this and in many just data analysis challenges more broadly is to do what's called dimensionality reduction. And so the idea is, as I've sort of alluded to, is taking high dimensional data and somehow compressing it or finding a low dimensional format of it. And so there's a few different reasons why one might want to reduce the dimensionality of data. One of them is, compression. So this is the sort of basis of things like MP3 compression or even JPEG compression with pictures, where you're basically, have many bits, you have all these pixels, and essentially the idea is how can you find a simpler representation. For instance, in JPEG, it's of edges within an image, and by forming those more efficient representations of the structures that are present, you can essentially compress the overall amount of data that you have to store. And that's how you get a smaller image out with JPEG, for instance. In addition, when we do dimensionality reduction, we can also benefit from the properties of denoising. And so essentially the idea is if there are core structures that we tend to see within data and we leverage those, then when there's noise or other artifacts within our data, we should also be able to effectively mitigate those or get rid of them just by solving this dimensionality reduction step as well. And then the third reason is also for interpretation. So in many cases, things like principal components analysis or PCA, which many of you might have heard of, perhaps in some of the other talks on AI as well, PCA and some of these other methods give us a way also of taking data and visualizing it and hopefully building some interpretation of this complex data. So I'm going to start by sort of providing a geometric picture of this idea of dimensionality reduction or of low dimensional models in general. And so we'll start just with visualizing this matrix here. So this could be sort of generic data where we're sorting features within the data along the rows. And then we have these different samples of our data points along the columns. And so in many of the examples that I'll show later on, we'll think about this as the samples being a point in time and the features being the activity of many neurons at that point in time. So essentially, we would have along each of these columns, we're going to measure the firing rate of, in this case, D different neurons that we can actually take a picture of. And then we're looking at how the activity of those D neurons is changing as a function of time. So that's moving along each of these rows in that case. And so we can now visualize each of these samples. So this D-dimensional representation of all of these neurons at a single point in time, we can think about that as a point in this high dimensional feature space. And the key sort of idea in dimensionality reduction and a lot of unsupervised learning as well within machine learning is that these points aren't scattered everywhere in this space, right? Oftentimes these structured signals that we see in nature, images, or in our case, neural activity, actually lies in lower dimensional subsets of the high dimensional space. And so the goal is really to find a way to approximate where those points live in the space, and then leverage that as a way of doing what I said before, compression and these other forms of interpretability as well. So what I'm showing here is just this picture where we have all these points, they're in this space, and now I fit this plane to the set of points. And so that gives me a linear approximation or a subspace approximation to this collection of data. And of course, you know, this same idea could also be visualized or thought about in sort of nonlinear ways. So instead of having to approximate all of your data with just this plane or this linear structure, you might actually try to find some curved or nonlinear structure to your data and use that as your model for the representations that are formed. And of course, we can kind of take this to even more exotic, you know, situations where when you start to think about really complex data sets where maybe different parts of the space live in different, you know, low dimensional subspaces. And this is a model that, you know, is now prevalent in a lot of work within deep learning and machine learning, where you might consider collections of objects, right? If I look at pictures of, you know, cats and pictures of dogs, it's likely that these data will actually consist of these mixtures of these low dimensional models. So this sort of sets the stage just for the introduction. I don't know if there's any questions just to kind of clarify this, this picture that, you know, we can take these high dimensional data and then we can try to find these low dimensional structures in them. I don't know if there are any questions. Okay.
No, por ahora bien.
Okay. No. Just wanted to stop if there was. Okay, so that sort of sets the stage. I mean, I think I'm not sure entirely how much you all have covered, you know, PCA or some of these methods already in the workshop.
We mentioned, but we didn't get deep to the principal component analysis, you mean?
Yeah. I was just kind of getting a sense of it.
Yeah, maybe it's good an explanation.
Oh, okay. Yeah. So, I mean, I can talk a little bit more here just about what principal components analysis is trying to do. So if you're familiar with PCA, essentially what PCA is doing is it's trying to find this yellow plane that I'm drawing here on the slide. So essentially you're finding the best low rank, meaning low dimensional subspace, and it's linear, so it has to be a sheet of paper and not this sort of non-linear structure that I draw on the second slide here. But we're going to find the best linear approximation to a collection of data under a loss that's just defined by a sum of squared error. So if you essentially pose this minimization problem as, you know, minimizing this loss between this plane and your data with a sum of squares, then that will essentially give you or converge to this solution of PCA. And the only difference that I mean, that's the high level. The only difference is instead of your actual data matrix, you look at your covariance and you then fit this. But the picture is the same, that essentially PCA is a way of finding this linear approximation to this collection of points. So maybe that at least provides a little context. And then the nonlinear version of this can, a popular method, if you all are interested in checking it out, would be isomap, which is a way of essentially, you know, finding this curve structure rather than assuming that you have a linear structure to start with. Okay. So that leads us to the sort of kind of core motivation of the work that I'll talk about in the next part of the talk, which is, you know, something that we encounter in many cases. So we've done this job, we've, you know, reduced the dimensionality, we found this nice, you know, embedding of these points in this, say, low dimensional space. And then we're often faced with a comparison question, which is how do two sets of representations or how do these distributions differ, right? And this could be a question of, you know, if I'm looking at, say, neural activity and I look across time, so perhaps, you know, some learning has taken place and now I have this new distribution and I'm like, I'm interested in how these two things relate to one another. Perhaps, you know, some disease process is underlying, you know, this, and maybe I have these two distributions that are across health and disease. And in many of these cases, I'm sort of interested in how these collections of data, or in our case, populations of neurons, are related to each other. And so we're faced with this question of how can we actually compare to underlying distributions. So here I'm just showing a very simple case where I have essentially the same shape, but I've just rotated this cloud of points, right? And so one way to potentially think about solving this sort of problem is by something that's called distribution alignment. And so essentially what I'm showing here on the bottom right is the sort of high-level strategy that we'll use, which is, so I'm coloring all the points here based upon where they are within this role, right? But the algorithm doesn't know which points are blue and which points are red. But the goal is to essentially bring these two things into alignment with one another, such that some notion of loss is minimized, right? So essentially our goal is to find a transformation such that we can basically map the red to the red and map to the blue to the blue. And so a mathematical formulation of that is shown here on the top, where I'm basically just, so mu is our source distribution. We're going to apply a transformation t to that source distribution in order to match our target distribution nu under some divergence measure d. So essentially, this is just the way of posing what I've said here in words of, you know, finding this transformation T that's going to bring these two clouds of points into alignment with one another.
Eva?
Yes.
Sorry, I'm Javier from Chile. Just to know better, is this just an isomorphism on the data that you're trying to pose?
Yeah, so that's a really great question. So there are, at this point, I haven't necessarily specified what the transformation class is. But yeah, you could imagine if I have, say, a unitary transform or something that just rotates the data, then I would essentially have an isomorphism between the data points. And I will show later, we have a solution for this where we just try to find regions. And in that case, you know, I think what you're asking about would be a special case of the general framework.
Okay.
Does that clarify?
Yes. Hello. Thank you.
Awesome. Yeah, so that's a great question, because the way I've written it out now, I haven't necessarily specified what this curly T is. So we don't yet, we haven't yet specified what types of transformations. So, okay, so the first, well, there's a few different design decisions that we can make, right, when looking at this objective function. So we have the distance, right? So, you know, what is this D? We have the class of transformation. So what should this, what types of transformations do we want to consider for T? And then also there's another part of this, which I haven't written explicitly, but we'll get to, which is if there's additional structure within the target or within my source distribution, can we leverage some underlying structure in that distribution as well? So the first thing that we'll kind of dig into is the first, which is the distance that we should use. All right, so so what I'll describe in the next few slides, unfortunately, I'm sorry, I'm not entirely sure of the audience and how deep we want to go into the math. If I mean, you're asking about isomorphism, so I feel like I can go a little deeper. But also just
well, the audience is just to clarify, Eva, the audience is quite variable. I mean, okay. You have from people that is very used to computer and machine learning and all this stuff, but also people that is just starting. So maybe, I mean, you can start and go as deeper as you want, because some people will follow you, some not, but it has happened to all of us in different aspects of the workshop, because
Okay.
nobody is, there is not so much people expert in everything.
Right. Okay. Well, feel
Oh,
free to ask questions
yeah.
again.
Okay.
I'll go into this with some depth
Okay.
then. Okay, so to answer this first question of what distance are we going to use, we're proposing to use what's called the Wasserstein distance, which is actually part of this sort of family of distances that can be used across distributions. And for now, I've just kind of written it up there as this new distance. Instead of the D, we're going to use this other notion of divergence. But I have a picture here below, which hopefully will give some intuition about it. And I mean, I guess I should also say that, for the types of problems that I've described, right, where you're interested in finding these matchings, this Wasserstein loss gives a much smoother and more geometrically sort of informed way of potentially finding matchings between different sets of points. So I'll give some intuition on the bottom. So the idea is, if I'm interested in, say, finding the distance between these different distributions on the bottom, I have a blue, I have a purple, and I have a red here. So because let's just say I want to find the distance between the blue and the purple, because the blue and the purple are actually not overlapping, if I were to use just a normal distance like just the Euclidean distance, or if I were to use a divergence measure like the Kale divergence, essentially because the support of these two, or the support of the two distributions doesn't overlap, they're going to look like they're orthogonal or completely dissimilar from one another. And if I look at the blue and the red, they're going to look just as far apart. Although if I do believe that there is some notion of geometry or sort of distance across space, then it might make sense actually that the blue and the purple are more similar to one another than the blue and the red, right? And so essentially what the Wasserstein distance gives us is it gives us a way of leveraging that notion of distance when defining the cost. So essentially what we'll try to do is we ask, how much energy does it take for me to morph the blue into the purple when my notion of cost is defined according to the ground distance that I have to travel? So in that sense, because I'd have to go a lot further to turn blue into red, the distance is much higher. And you're seeing here on the bottom right this idea that of the Wasserstein distance, right, that blue and purple are kind of close, and then blue and red are much further. So this is codified, or can be written here using this equation. So this is now just computing the Wasserstein distance between two distributions, P and Q. And what I'll do is I'm just going to say the distance is defined by essentially solving this optimization program to find what's called a transport plan P. And this transport plan is going to tell me how I'm going to transport mass from P to Q according to some cost that's defined by, it can be defined by a number of different things, but in this case, I'm just saying, what is the distance between these two bins within this space that I showed in the previous. So for instance, if I just measure the cost of moving across space with an L2 distance, or mean squared error, then I essentially would get to that picture that I showed in the previous example, where something that's further away is much more costly. And so essentially you're multiplying all these costs by the entries of this transport matrix. And then the other piece here that's important is this, okay, so you also have this constraint on your transport plan, namely that it marginalizes to your source and your target. So that's just to say that under this transformation, you will get from your source to your target. And then the last part here is just an entropy term, or a negative entropy in this case. But what it's going to do, because we're minimizing, is essentially smooth this transport plan that you're trying to find. So this is a visualization. On the rows, I have some distribution, a P, and I have a target Q. And what I'm visualizing is the transport. So how you would actually move mass from one part of the space to match the other. And then as we move to the right, each of these matrices is showing different levels of smoothing. So if I increase the amount of entropy, I'm essentially taking what is at some point this kind of pencil curve, and I'm spreading it out with more entropy. So what that effectively does is it forms a type of regularization, so you don't have to have as stringent transport, and you allow a little bit of wiggle room to the program. Importantly, it actually allows for computational efficiency as well by smoothing things out. So that's also another major advantage of using optimal transport. So the first part of our solution that I've described is which distance we should actually use for solving this problem. And then to answer the next two questions, we're going to go to our specific application as a kind of guiding application to motivate some of the answers to number 2 and 3. So I will actually dig into the neuroscience now. So this is the kind of, as I said, motivating application of neural distribution alignment. So what I'll show now is an example where we are recording the neural activity from a non-human primate while from their primary motor cortex while they're carrying out a center out reaching task. So basically what that is they're holding a cursor, there's a screen that they're looking at, and there's going to be basically this yellow dot that's going to go either to the center and then go to one of 8 different targets around the center. And then the animal's been trained to basically make a movement towards the particular direction that's illuminated under the cursor and then come back. And so what I'm showing over here on the right is an example of a movement distribution that could be carried out while they're reaching towards, say, 4 different targets. And so this is now plotting the velocity in X, the velocity in Y, and the different colors represent different reach directions. So if they're going, towards a particular target, that's color coded. And then each of these points in space is going to be a specific instant in time. And so this is sort of this movement distribution then that we're interested in taking neural activity and understanding whether or not we can actually align this to an underlying movement distribution. What this will allow us to do, if we can do it, is essentially solve this decoding problem of mapping neural activity to outputs, right? Or to, in this case, specific movements that the animal wants to carry out, which is, in terms of applications, a way forward in being able to do brain-machine interfaces or brain-machine control. So essentially, one of the reasons why we can actually do this, or why we know that this is possible, is because we know that when we take the activity of neurons while animals are carrying out different movement tasks, the structure of the underlying movements is actually embedded or preserved within these high-dimensional neural activity patterns. And so if we apply something like PCA, or in this case, a factor analysis technique to this neural activity, we actually find that we get the shape of these movements within this brain activity pattern. And so our question is whether or not we could actually find a way of using distribution alignment to bring these two things together. And so we do that by I guess, one of the key insights that we do leverage in this formulation is along the lines of this third bullet that I had earlier, which is what is the underlying structure of this data or these distributions, and can we actually use this to then inform a solution of sorts? So in this case, we can leverage the fact that these distributions actually consist of clusters, and there are actually many different applications, right, that could also fit this sort of structure. So in this case, we can use the fact that we know that data is clustered to try to help with the alignment procedure. But then I'll just remind you that we know that they're clusters, but in our case, we estimate them and we don't know which clusters correspond. So we don't know, which clusters are green, and we don't know which ones are red, for instance. And so what we do is we can take our earlier optimal transport alignment problem, and we can actually decompose this into this hierarchical sort of formulation. So in particular, what we do is we estimate clusters within the data set, and then this inner part here that I'm showing is essentially forming alignment or finding a transformation T, a local transformation Tij that aligns cluster I with cluster J under this Wasserstein loss. We can do that for all these different pairs of points. And then we're essentially going to solve this transport problem to now match the different clusters to one another by this outer Pij term. So essentially what we're doing is we're doing transport between clusters simultaneously with transport between the points within these clusters. And again, we add this entropy term. And to get back to the earlier question, in this case, we assume that T lives on, is a unitary transform, and so preserves geometry and can rotate and can basically just rotate the points, but can't actually stretch or scale them in different directions. And so what we do is we can solve this problem locally, which is nice because there's some computational benefits. And then this outer portion here that I'm showing on the right is essentially a constraint now on the fact that you should have a global transformation T where you have consensus across these different Tijs, that they should all agree on the same global transformation. So yeah, this is based upon, these results and solution are from a paper that we had at last year's NeurIPS. The kind of key contributions of this work were this novel hierarchical formulation. We have a distributed sort of solver. And then I'll show you, we also were able to improve performance within this neural distribution alignment problem. So here's just some examples. First, on synthetic data, I'm just showing now, so our method is called HIWA for hierarchical Wasserstein alignment. So that's the blue and the yellow curve on top. And then below, we're showing in green the Wasserstein alignment. So that's just if you use optimal transport to solve alignment, but don't use the clustering structure within your data. So what we show here is that leveraging this clustering structure using this hierarchical formulation is really beneficial in being able to improve our probability of getting alignment with these sorts of methods. Also, just to sort of show an example in a also sort of synthetic case, but where we take now our movement distribution and now we rotate it with a fixed rotation, and then we can compute the Wasserstein or the KL divergence between these different clouds of points. And so what I'm showing here, so the optimum should be at 0. And so we see that all of these different types of regularization for the entropy and also the KL all converge at the same point, or also all have a global minimum at the same point. But what we see is that, for instance, with this blue curve on the bottom, using optimal transport with some smoothing, we get a much smoother landscape in comparison to, say, the KL divergence above, which is really important to note because all of those little bump, all those different points above on the red curve are local minima, and it introduces, a lot of places where optimizers can get trapped in. So this is just to show that optimal transport gives us also this nice smooth landscape. Okay, and so now this is showing results now on neural data. And so what I'm showing here on the left is an example where we take activity from motor cortex within, one animal, We embed it into 3 dimensions, and that becomes our source distribution. We align it onto this target in the center in order to get this aligned version on the bottom. We can do this again with a different animal performing the same task and find actually alignment onto the center target as well. So in this case, it's kind of interesting because I guess there's 2 things to point out. One is that, you know, even though these are 2 completely different brains, 2 completely different sets of neurons that we've measured from, because the behavior and the movements that are sort of constraining this neural activity are quite similar, we actually find that we can solve this across subject alignment problem, which means, you know, in the future, or kind of taking this maybe to a different level, we could potentially, you know, just record neural activity from one individual's brain. And if we knew something about what they were doing or the behavior, it could be potentially aligned onto a different individual's, say, target distribution or say movement or what they're watching or what they're viewing. And so this could give you a way of potentially solving across brain alignment problems in the future. So that's kind of cool. But of course, as I said, it's sort of constrained with this specific behavior. And I'm just showing here on the bottom through a few metrics, just that, you know, if we measure the nearest neighbor error, which gives us an estimate of whether or not the target is aligned, we find that after alignment, we can get a pretty good agreement, at least with the different reach directions, with, you know, some small amount of error. So in general, we find that the correct targets can be, or sorry, the correct clusters can be aligned to one another in this case.
Sorry, Eva. What do you mean with this? That knowing if you have information about the activity of one individual, and you have some information of the activity of another individual, and the information about the target, does mean that if you make a first experiment in which you have a kind of the relationship between both brains in response to the same target, then you from one you can extrapolate what may be occurring in the other brain later in another experiment. You mean if I not if
yeah.
No,
that's a great question. Yeah, that's it's good to clarify. So in this specific case, for instance, you could, you know, do as you said, which is in subject 1, I actually know the correspondence, right? Potentially, well, actually, I don't even actually need to know the correspondence. I'm solving that as well. But I know that what the green points mean, right? Like, I know that means move a controller over towards the left, right? And I know that the points, you know, on the right correspond to kinematics of this other direction, right? So when I essentially take this source distribution, I place it onto the target, I can now give the labels that are on the target back to the source, right? So so essentially you're
kind of reverse engineering the problem. I mean, no, it's kind of.
I mean, in some sense, if you were interested in how 2 brains are related to each other, this could give you a way, at least, of constraining things such that you could essentially go between the 2 brains through this underlying latent space, right? And if the latent space, or if the key things that are driving these activities are similar, you could, you know, presumably use this sort of idea and approach to tackle that problem. And in this case, what I'm showing is not aligning 2 brains to each other, right?
No, it's just,
yeah, but you could imagine kind of almost being able to do that at some point, as long as the latent spaces are sort of constrained. And so it would have to obviously be under certain caveats. But we're working on it, so or we're trying. Okay, so this is now another example. So this is now in rat cortex. Oh, sorry, this isn't in cortex. This is in striatum. And what I'm showing here is now, instead of looking at, the different colors being different reach directions, I'm now looking at different time points as being in different either sleep states or the animal being awake. So we have REM, non-REM sleep, and then also wake color-coded. And on the left, I'm just showing, if I just take a block of data, 12 hours, I do this dimensionality reduction. In this case, it's just with PCA. Now I look at this distribution. I can do the same thing also for a different block of data, but all I'm showing is, while you have some kind of core structures that are similar across both because of these sleep sort of states being aligned, the low dimensional projections of them are not, aligned, right? So it's not like I can say, oh, being in this part of this space corresponds to being awake. But if I can actually bring these two things together, then I can say, okay, when I'm in this part of the space, that's what this means, right?
Sorry, in this case, the source and the target, the source is the activity, the neural activity, and the target
is also neural activity, but now just from a different time point.
From a different time point.
Yeah, so now we don't have a target distribution anymore. I mean, we have labels, but we don't know what the shape of, sleep versus not sleep should look like.
So these are the source and the target, two different times, two different activities, neural activity in two different times.
Correct.
Okay.
Yep. Okay.
El mismo individuo, the same individual as in
individuals, and presumably it's mostly the same neurons, but it's over a pretty long time period, so it's likely that the spike sorting, might have some flips and some permutations. In general, we do our best to try to match units, but this is also an approach because we're trying to align latent spaces. We don't necessarily have to have the units align, right? You can have a set of neurons from one day and a different set from another, but you do have to assume that they're encoding similar pieces of information. If they're completely different, then, all bets are off. So what I'm showing here along the top is just, now if we take what I showed on the previous picture, and now I basically bring these two data sets, such that they are aligned with one another. So now I'm taking A and B, and I believe I'm aligning A to B. And so, yeah, so in this case, you can now actually get a very nice picture of how these two things are related to one another. And then on the bottom, I'm just showing a sort of a simple competitor method called CCA, canonical correlations analysis, which is sort of like a two data set equivalent of PCA, where instead of just finding, the subspace for one set of points, you're finding a common latent space that sort of is maximally informative about two data sets. And so all I'm showing here is just, if you apply CCA, even if you maximize the correlations, you're not guaranteed to find this nice alignment of your different clusters or data points. Okay, so, and then again, I'm showing some metrics to kind of quantify that and show that, with this method, we're still, at least if we look at over here, the nearest neighbor error, for instance, we can get much lower with HIWA, and then with CCA, the accuracy is degraded. And also visually, you can tell that these things are true. Okay. So, and then I know we started a little late. Are we ending right at 4? I mean, I want to make sure there's time for questions too. So maybe I take another 5 minutes. Okay. So in the last part of the talk, I wanted to kind of talk a little bit about how we might, revisit this sort of earlier objective, which was, throughout I've been talking about, we take this high dimensional data and we've applied some dimensionality reduction. Once we have that, we have this latent space. And now I've talked about how do we align sort of representations and distributions. And so the natural question would also be, what if we don't already have this known nice embedding? Is there some way that we can actually solve this whole thing in an end-to-end manner? And so that's what we've been working on more recently, and leveraging deep learning in this case to be able to tackle this problem. So it's just really a teaser at this point, but essentially our goal is to basically solve both for this low dimensional embedding along with this alignment objective. And so what we've done thus far is essentially, training just a deep learning model, which is an encoder, to take this high dimensional neural activity, embed it in this low dimensional space. And our objective is essentially a multi-task objective. So on one hand, we're interested in aligning the points using this Sinkhorn or this Wasserstein alignment objective. So in the case where we have a target, right, we can kind of specify a similar objective as what we had before. But in addition, in order to further constrain this encoder, we also ask the system to be able to do a number of other tasks, like be able to classify or between the red or the orange and the green, and also to be able to reconstruct the neural activity through a decoding path as well. So essentially by combining many of these objectives together, we can actually start to make progress towards a fully end-to-end solution. And so what I'm showing here is just deep distribution alignment across multiple iterations. So just starting with, these kind of raw projected points that really don't have a whole lot of structure at the beginning of training, and then converging over time towards having something that is actually, both a very good low dimensional projection and also aligns well with the target distribution as well. So then kind of preliminary, but we're really excited being able to tackle all this. And going back to Maria's earlier question, I think in order to really tackle some of these hard across subject alignment problems, being able to use nonlinear structures is likely to be really critical. And so we think that deep learning provides a way of, tackling this using this nonlinear function approximation. And so with that, I'll wrap up. I talked about at the beginning how dimensionality reduction gives us this way of finding low dimensional representations of data and neural data sets in particular. I introduced a new method, and we have code for this available as well. If anyone's interested, please reach out. I think I have some links later. This hierarchical optimal transport method, and I showed examples of it in decoding movement and also decoding sleep and wake states in rodent. And then finally, a teaser of our solutions for deep distribution alignment to solve this problem in an end-to-end manner. And then I'll acknowledge, so students in my lab that have worked on this project, Max DeBeja and Mehdi Azabou, and also our collaborators, so Keith Hengen and his student at WashU, and John Lee at Georgia Tech, and Chris Roselle, and then our funding sources. And then, so here on the bottom, I just have this link to this website, Neuroline, and we have code and some data sets and some other things there if you're interested. So thank you so much. I'd be happy to take any questions.
Thank you, Eva. Questions, preguntas?
I have a bunch of questions.
Good.
All right, thanks for the talk. It was great. I think I thought I was understanding up until the point in which you introduced the deep learning. So there's some things. First, like how much? What's the amount of data that you have to train these neural networks? Or I mean, what's the data you have?
Oh, so it's actually not that much data. There's maybe between 500 or 700 time points, at least just for this simple data set here. But because it's all unsupervised, we're basically just using the whole data set to train. And there's no testing in that case, right?
Yeah.
But yeah, no, surprisingly, I mean, it is a very low dimensional task, right? So we know that in 3 dimensions, you can preserve a lot of this information. So perhaps, despite the conventional wisdom of needing a lot of data, you don't necessarily need too much for this, at least.
And you have like three dimensions. I thought they were like more. So when you say like higher,
no, I'm just saying the underlying structure is pretty low dimensional. So we have like 150 neurons or so. So we start from 150 dimensional space. We're going down to say a 3 or 2 dimensional space. And I'm just saying, because it is so low dimensional, we don't end up, I guess, needing as many data points.
Okay, yeah. So, and then so what the classification part of the task, in which you have like so I come from the more like machine learning side, so I don't know a lot about neuroscience. So what are those different colors represent? What are the categories you have, your output category?
Yeah, so in this particular example, all I'm just showing is this center outreaching task, right? Where this animal is making movements to a particular reach direction. So essentially each color is a different target that's around a center point. And essentially the idea is that, different neurons are encoding these different directions in different ways. And so essentially you get this sort of clustering in this neural space that is also sort of aligned with the different clusters that you see here in the movement space. Does that clarify?
Yeah.
And then the other example, I was showing clusters where it was, sleep or not sleep.
Yeah. Okay.
It can kind of be a general approach, but in this case, it's reach direction.
It's like the real world counterpart. What's going on, I guess.
Yeah.
Yeah, I have more questions, but I will let anybody else like.
Well, also feel free to shoot me an email if you have more questions. I'd be happy to chat.
Okay, awesome, thanks.
I do have 2 questions, but I guess I'm going to focus on one first, and then if there's some time, should the another. But how do you do? And this is more abstract, broad question, but how do you do when you don't know the structure of your data a priori? Because you mentioned manifold learning or something like that, but how do you, because this decision on transforming, on giving a distance from your data to a desired distribution, you somehow need to know what is the shape of your data. So how do you do it in the first place?
Yeah, I mean that's a very, as you said, a very kind of big question. I mean, I think that there are many applications and many cases where, assuming that you have clustering in your data, perhaps just because you know that you have these different classes, right? Or that there should be differences across different points. I feel like that's, a pretty reasonable assumption in many cases, but even then, you don't necessarily know. Even if you have multiple classes, maybe some of them are actually kind of part of one, kind of larger manifold, right? With that being said, obviously if you have an assumption like, is my data clustered, there are ways of checking that assumption. For instance, you could fit a mixture model to your data and you can ask questions about, the fit and the accuracy. But yeah, I mean, if you don't know, okay, so that's one thing, and maybe that's a little bit easier. But then your other question is, what if it's some sort of, I don't know, weird structure that you don't necessarily have a handle on even. And I think that's really what makes unsupervised learning very hard, is that without additional assumptions or labels, you're sort of, left to learning what the structure is. And then you kind of have to see how these different assumptions, better approximate or better predict new data points that perhaps you know, could fit better with one or the other kind of assumption. I don't know if that really, I mean, that doesn't really answer your question. I mean, obviously, if you make an assumption about whether or not your data has a particular model, you can at least get some measure of goodness of fit, right? Mm-hmm. But yeah, I mean, often, you know, people try to visualize things in 3 dimensions just because we don't know how to know what structure is present in our data in higher dimensions, right? So it's a pretty fundamental question.
Okay, thank you.
You're welcome. Hopefully that was helpful in some way, but very meta.
More questions? ¿Más preguntas?
And certainly I'll just say, you know, to test the assumption that your data has a low rank approximation, there are very well established tools, right? For instance, using the singular value decomposition and then looking at, you know, how the singular values of your system are decaying can often give a lot of insight. And even just looking at kind of interpoint and these distributions of distances across data, which is ultimately what's being leveraged with these manifold learning techniques, right? Like that can give you some insight too.
Mm-hmm.
Cool.
Okay, and in this way, this way of analysis is informative in order to kind of improve the artificial networks for analysis.
Yeah. No, it's so we are now embarking on a journey into that as well, because as you see, like this idea of distribution alignment or looking at distributions, right, could be applied pretty broadly. And so you could also imagine applying these same techniques in looking at representations formed within neural networks, which is what we're doing now, and using a lot of these same tools and machinery to then try to understand, like how neural networks are working, or when they will be performant, or when they'll be able to learn effectively as well.
So and in some way has a kind of predictive power. I mean, you have a kind of the possibility to predict the efficiency of the neural network, artificial neural network.
Well, that would be the hope. I mean, so part of it is like asking questions about how architecture and how different parameters, right, give rise to better learning outcomes. And you can use these sorts of tools in representational geometry, as it's sort of called, to get a handle on some of those questions. And that is an area of research now, where people basically look at these manifolds as formed in neural networks, and then they look at some of these aspects that I kind of talked about earlier on as well. So it's fun. It's really fun times for neuroscience and artificial intelligence. Things are converging very quickly. And I think the fact that, you know, modern machine learning is leveraging neural-like architectures is giving a lot of nice points of convergence. So yeah, it's pretty cool. And it's good that you're holding a workshop on these topics.
Yes, in fact, we are just in the very first steps of moving in this direction. Pushing a little bit, pushing. The feel is for the young people.
For us all. It's for us all.
Yeah.
All right.
Okay. So more questions, más preguntas, comments. Please. This is the time, the correct time. Este es el momento. Ahora nunca, now or never. No, not never, no. Because
not never.
Not never, no, I know. Eva offer Eva lo que propuso es que cualquier otra pregunta que tengan o comentario que le pueden escribir un mail. So you may email to Eva. She's very open to receiving news about questions and comments. Acá dicen, muchas gracias, Eva. I get a lot to think. Elizabeth. And so, well, thank you very much. It was really a pleasure to have you with us, and very inspiring talk and very challenging talk also. So, yeah, sometimes you need to have some high level talks, because this makes you kind of eager to become eager to improve.
Yeah, good. Cool. Well, thank you so much for having me.
Thank you so much.
Take care. Bye, everyone.
Thank you. Goodbye.
Bueno, gente.